Welcome back, fellow mitochondriacs, for an episode of Cancer is a Mitochondrial Metabolic Disease. Today, I'm going to discuss a recent paper that was recently put out in November of 2025. And the paper was highlighted by a video that was published on December 18th, 2025, by a PhD YouTube influencer. To be fair, I generally really like the videos that this individual puts out, so I'm going to keep it kind. Despite my initial anger about the clickbait title that was used, which scared much of the audience and which prompted multiple emails to be sent to me regarding the video, I'm not going to say anything negative about the person who put the video out. I do think, however, that if you're going to publish a video with a clickbait title that is clearly meant to scare people or confuse people at minimum who are going through one of the worst times of their life and who are fighting for their life, I would ask that you do it with more sensitivity and care. I also think that the entire story or the whole story regarding the paper should be told, which is what we're going to do here. So without further ado, let's get into it. So I have decided to break this paper down into sections. And in all likelihood, this will likely be a couple of videos just so we don't get overwhelmed, because I've made a response of about 90 PowerPoint slides. And I really wanted to be thorough with this paper and give thorough, thoughtful, science-based rebuttals to each of the sections of the paper. And the paper is actually fairly long and they did a lot of experiments. So I want to make sure that we can hit everything. So the first section will be intro into the new scary paper. So the paper was published November 4th of 2025, published in Cell Metabolism, and it's titled Induction of a Metabolic Switch from Glucose to Ketone Metabolism Programs Ketogenic Diet-Induced Therapeutic Vulnerability in Lung Cancer. And by the title itself, you would not be thinking that this is a hit piece on ketosis, but it is. So in the abstract, it says tumor-initiating cells, TICs, preferentially reside in poorly vascularized, nutrient-stressed tumor regions. Yet, how they adapt to glucose limitations is unclear. We show that lung TICs, tumor-initiating cells, unlike bulk tumor cells, can switch from glucose to ketone utilization under glucose deprivation. Ex vivo ketone supplementation or a prolonged ketogenic diet supports tumor-initiating cell TIC growth and tumor-initiating capacity. Integrated metabolomics, genomics, and flux analysis reveal that ketones fuel ketolysis, fatty acid synthesis, and de novo lipogenesis. Paradoxically, ketogenic diet intervention creates metabolic vulnerabilities in TICs, sensitizing them towards inhibition of ketone transporter monocarboxylate transporter 1, regulated by its chaperone protein CD147, as well as towards pharmacological blockade of fatty acid synthase. Loss of CD147 ablates TICs under glucose limitation conditions in vitro and in vivo. These conditions uncover a nutrient-responsive metabolic switch in lung TICs, tumor-initiating cells, and provide mechanistic insight into how dietary manipulation can influence cancer progression and enhance efficacy of targeted therapies. So in that abstract, there are some things that we know about already. There are things that would be potentially considered bad, and there are some things that potentially could be seen as a win. And we're going to go over each of these. Well, first of all, there are these bulk tumor cells, and then there are these TIC initiating cells. And basically, they're trying to differentiate between the two and the capabilities that each of these two have within this lung cancer model. And they're effectively saying that these TICs, these tumor-initiating cells, they can switch, they can be more metabolically flexible, they can up machinery and make fatty acids. And it kind of illustrates here that the acetyl-CoA broken down from ketones will be going into the mitochondria to make energy as well. And then it also talks about these MCT1 transporters, the transporters that transport some of the ketone bodies into the cancer cells. And by blocking that can create a vulnerability. So basically they're saying if you put them on a ketogenic diet and then you block these transporters, then that can effectively, potentially kill these TIC cells. So the introduction, metabolic reprogramming is a hallmark of cancer, enabling tumor cells to meet increased biosynthetic and energetic demands. Most tumors exhibit dependency on glucose and glycolytic flux or glycolysis, upregulate glutaminolysis and nucleotide synthesis, or may utilize alternative carbon sources such as lactate. These alterations sustain tumor proliferation, invasion, and maintenance of stem-like properties. Otto Warburg first discovered that tumor cells exhibited high rates of aerobic glycolysis and proposed impaired oxidative phosphorylation as the underlying cause. However, mitochondrial OXPHOS defects are rare in most cancers. Isotope tracing experiments in several cancers demonstrated unequivocally that glucose was oxidized to carbon dioxide in tumor cells. Quite strikingly, tumor cells display metabolic plasticity through balancing glycolysis and OXPHOS in response to microenvironment conditions. Under normoxia, normal oxygen conditions, high rates of aerobic glycolysis and OXPHOS provide biosynthetic precursors and ATP to fuel anabolic processes essential for rapid cell growth and proliferation. On the other hand, hypoxia-inducible factor 1 alpha drives glycolytic enzymes while suppressing OXPHOS during hypoxia. I'm going to leave the commentary for when I give my rebuttal. I'm just going to read it as it is. Beyond glucose metabolism, non-small cell lung cancer, NSCLC, tumors exploit lactate and other metabolites as fuel. Lactate, exported via monocarboxylate transporters, monocarboxylate transporter 1 and 4, leads to the recycling of nicotinamide adenine dinucleotide, NAD, for glycolysis and serves as a carbon shuttle to support the tricarboxylic acid cycle and lipogenesis. Nutrient exchanges between tumor and stromal cells, such as alanine provision by pancreatic stellate cells, further highlight how metabolic plasticity enables tumors to overcome nutrient limitations. Tumor-initiating cells, TICs, a subpopulation driving tumor initiation and relapse, exhibit distinct metabolic dependencies, including reduced glycolysis and elevated reliance on exogenous methionine. Given that TICs tend to reside in hypoxic or poorly vascularized regions of the tumor, they may activate hypoxia-inducible factor 1 alpha and pyruvate kinase isoform type M2, PKM2, to sustain glycolysis, or utilize alternative carbon sources such as glutamine and acetate for lipid biosynthesis. However, how TICs adapt under glucose-limiting conditions has remained unclarified. Here, we showed that TICs, tumor-initiating cells, overcome glucose deprivation by activating ketone utilization. Unlike their differentiated counterparts, TICs relied on ketones for growth under low glucose conditions, a dependency reversed by glucose restoration. Ex vivo supplementation of ketones or prolonged ketogenic diet demonstrated ketones to be essential for maintaining TIC properties. Paradoxically, the ketogenic diet induced dependency towards ketolysis and sensitized TICs to MCT1 inhibition. Ketones also fueled de novo lipogenesis, or the making of new fats, thereby enhancing the anti-tumor effect of fatty acid synthase FASN inhibitor under a ketogenic diet. CD147 was found to be a critical chaperone regulating MCT1, and its loss ablated lung TICs under glucose deprivation. These data revealed a hitherto undescribed metabolic switch in lung TICs and demonstrated how dietary intervention could modulate cancer progression and therapeutic responses in a context-dependent manner. So I'm going to be using Dr. Seyfried's paper that came out in April of 2025, as well as other papers that are important to break some of these things down. So the first portion I'm going to talk about is a questionable assumption, number 4 as part of his paper, but a questionable assumption that was made also in the paper that we're describing. In fact, I'm going to go to it right now. It says, however, mitochondrial OXPHOS defects are rare in most cancer types. So I'm going to go to the response here and say that based on the foundational principle of evolutionary biology and in recognition that mitochondrial structure determines function, the information in Figure 2 and Table 1 present substantial evidence for abnormalities in number, structure, and function of mitochondria in all major cancers. Moreover, no tumor has yet been described with normal content or composition of cardiolipin, the intermitochondrial membrane-enriched phospholipid essential for the efficiency of OXPHOS function. Reductions have also been reported in neoplasms for mitochondrial coenzyme Q, which, like cardiolipin, is also essential for OXPHOS efficiency. Hence, these findings considered collectively address the premature criticisms of Winehouse in providing a significant measure of support for Warburg's central hypothesis. And what this paper shows is not only a table of all the cancers that have observed mitochondrial abnormalities, number, structure, function, but it also gives electron micrographs to prove that the cristae are disorganized and or not normal. And I wanted to highlight in red here that lung cancer is one of the cancers that has known by multiple authors and in multiple times, multiple years, publications showing dysfunctional mitochondria within lung cancer. Now, it doesn't say small cell versus non-small cell. I'll give it that, but this is lung cancer, and these mitochondrial defects are there. Another thing that we're going to talk about in the future under a next section under hypoxia versus normoxia, they're saying that under normoxia with normal oxygen, high rates of aerobic glycolysis and OXPHOS provide biosynthetic precursors in ATP to fuel anabolic processes. On the other hand, hypoxia factor 1 alpha drives glycolytic enzymes while suppressing OXPHOS. So they're saying when they're hypoxia factor would be upregulated, which drives glycolysis and glutaminolysis, I might add, suppresses OXPHOS during hypoxia. It's also saying that the cancer cells that are the most likely to use OXPHOS and ketones tend to reside in the hypoxic or poorly vascularized regions of the tumor, and they may activate hypoxia-inducible factor 1 alpha. So they're saying they're activating HIF1 alpha, which we know shuts down OXPHOS and upregulates glucose. And they're saying that the TICs are not utilizing glucose and they're utilizing OXPHOS. But we'll talk about that when we start talking about hypoxia. So this is Seyfried's newer graphic that came from their paper, I believe I want to say this is the April of 2025 paper. It's very similar to an older graphic they had, but it is nicer and a little more complete. And so the argument that is made by Seyfried is that after a transformation event, the amount of OXPHOS used by a cancer cell diminishes as the malignancy progresses, right? So a less malignant but cancerous cell would have some ability to utilize OXPHOS, whereas the more aggressive and malignant phenotypes would less likely to use OXPHOS. And that's been seen in multiple papers. Generally, the more aggressive and metastatic a cancer is, the less OXPHOS they utilize and the more SLP they utilize. So there's going to be some going on. That is true, but it's insufficient for maintenance of that cell to survive, proliferate, and grow. You'll also notice here that there's a gradual decreasing of the cristae, and as these cristae start to become more disorganized, then you'll have an increased reliance on SLP and decrease OXPHOS and insufficient OXPHOS ultimately toward the end of this here. This is corresponding with this graphic here. And then these little graphics here, the electron micrographs are showing a normal mitochondria with normal cristae and then like a ghost mitochondria which has almost no cristae formation. So these cristae are critical. As this paper is titled, Mitochondrial Cristae Shape Determines Respiratory Chain Supercomplex Assembly and Respiratory Efficiency. So it says respiratory chain complexes assemble into functional quaternary structures called supercomplexes within the folds of the inner mitochondrial membrane, or cristae. Here we investigate the relationship between respiratory function and mitochondrial ultrastructure and provide evidence that cristae determines the shape and stability of the respiratory supercomplexes and hence mitochondrial respiratory efficiency. And so what we're seeing here is that if you have a nice folded cristae, the mitochondrial supercomplexes can form and there is efficient electron transport. And if you have perturbed cristae or even lack of cristae, then these complexes are not going to form and they're not going to have efficient ATP utilization. And that's exactly what's shown here. So we lose the supercomplex, we have inefficient electron transport, decreased OXPHOS, this is well known. Then supercomplex formation, you have more oxidative stress because the electrons are not being pushed through the system like they're supposed to. And you have oxidative stress that leads to membrane peroxidation. And then membrane peroxidation further damages the cristae formation. Further loss of supercomplex formation leads to a vicious cycle. So we know that when we're on this side of the graphic here, on this side where we have increased SLP, decreased OXPHOS utilization, these cristae are not like they normally are. These are disorganized. They are not forming the respiratory supercomplexes like they should. They are becoming less efficient and they cannot put out energy like they normally would. This is irrespective of oxygen content. So this is just a blown up situation. So as we gain more mitochondrial defects over time, It's another way of looking at it is mitochondrial heteroplasmy. Basically, we have decreased amounts of OXPHOS being able to be utilized and a compensatory mechanism, substrate level phosphorylation, the fermentation of glucose and glutamine as being the backup to that system. To look at it from a different perspective, this is a non-tumor oxidative cell. It is metabolically flexible. It can use glucose, it can use amino acids, it can use beta oxidations and ketones. But in order for that to be the case, you have to have normal mitochondria, or at least functioning mitochondria. You have to have oxygen present, and that's going to make the vast majority of the energy under aerobic conditions. As we get to like this point here in the middle, we're going to have these cells that are oxygen consuming phenotypes where SLP is starting to upregulate itself to make up for the differences lost in the ineffective ATP generation because of mitochondrial defects that are accumulating and leading for these mitochondria not to be as efficient in making energy. So the SLP, the substrate level phosphorylation, will upregulate as a way to compensate for that. Ultimately, what we're left with, severe mitochondrial defects, severe cristae disorganization, respiratory supercomplexes cannot form. Therefore, there's inefficient or even very low ATP that's being able to be made. Again, this is irrespective of oxygen. Oxygen can be present here. It does not matter because the mitochondria, they are not able to make energy under these conditions. Now, if hypoxia is present, it even exacerbates even further. But this is irrespective of oxygen amount. And so what happens is the dominant form of energy production by these cancer cells is substrate level phosphorylation, glucose turning into lactate and glutamine turning into succinate through the mitochondrial substrate level phosphorylation. This we've been talking about for a long time. Now, this is a graphic that I don't show as often. And this is when we have disordered mitochondria that are not able to make energy, the cell will try to survive. And so, of course, glucose will be upregulated, glutamine will be upregulated, but so will things like autophagy, things that we always consider good, but are not always good. Autophagy and mitophagy are hijacked by cancer as a way to get sources of energy through amino acids, glucose, et cetera, et cetera. And then beta oxidation will also be upregulated a lot of times, which is the breakdown of fatty acids. Now, these fatty acids cannot be utilized, unfortunately, because we don't have a functioning mitochondria, and they cannot be fermented, which we'll talk about also in a future section. So let's then talk about normoxia versus hypoxia. All we've looked at this point is we've looked at structure and function. Now let's talk about hypoxia versus normoxia. So I pulled this paper that was basically defining normoxia, physioxia, hypoxia in tumors, implicating for treatment response. It was published in March of 2014. I'm going to read the abstract. Tumor hypoxia is increasingly recognized as a major deleterious factor in cancer therapies as it compromises treatment and drives malignant progression. This review seeks to clarify the oxygen levels that are pertinent to this tissue. It is argued that normoxia, 20% oxygen, is an extremely poor comparator for physioxia, i.e., the much lower levels of oxygen universally found in normal tissues, which averages about 5% oxygen and ranges from about 3% to 7.4%. This is not talking about the amount of oxygen in our lungs or in our arteries. This is actually in the tissues once perfusion happens. Importantly, it should be recognized that the median oxygenation in untreated tumors is significantly much lower, falling between 0.3 and 4.2% oxygen, with most tumors exhibiting median oxygen levels of less than 2%. This is partially dependent on the tissue of origin and is notable that many prostate and pancreatic tumors are profoundly hypoxic. In addition, therapy can induce even further, often unrecognizable changes in tumor oxygenation that can longitudinally increase or decrease depending on treatment in ways that are not always predictable. Studies that fail to take cognizance of the actual physiological levels of oxygen in tissues, approximately 5%, and tumors, approximately 1%, may fail to identify the real circumstances driving tumor response to treatment and/or malignant progression. This can be particular importance in genetic studies in vitro when comparison to human tumors is required. So let's see if we can get organized here. So the percent oxygen at sea level is 21%. And then the amount of oxygen actually in the alveoli of the lung is about 13.5%. And then what actually gets into the bloodstream, into the arteries, is about 9.5%. And then at the end of circulation, by the time it gets back to the heart, effectively, it is about 6.5%. However, at the tissues, it's quite a bit lower than that. So physioxia, or the amount of oxygen in the tissues, to be physiologically healthy is saying there's an average of about 6%, ranging from 4 to 7.5, depending on the tissues. 5% is a proposed compromise since it's often used. It's saying physiologic hypoxia would be at 2%, the low level of which normal hypoxic responses are listed, lower limit, approximately 1%, upper limit, less than 5%. And then pathologic or disease hypoxia shows persistence of poor oxygenation, suggesting disruption to normal homeostasis. Below this level is pathologic hypoxia applies, 1% or below. And then radiobiologic hypoxia, the oxygen level in which cytotoxic effects of radiation is half maximal. And we'll talk about that at length when we go into oxidative therapies. But I'm just going to digress for a moment and just talk about specifically the amount of oxygen in tumors by tumor type. Before I go there, though, I pulled this paper from July of 2009. It's titled Changes in Oxygen Tension Affect Cardiac Mitochondrial Respiration Rate via Change in the Rate of Mitochondrial Hydrogen Peroxide Production. It says, in contrast, sustained incubation of mitochondria at a PO2 of 10 to 13 millimeters of mercury for 30 minutes caused a 50% decrease in mitochondrial respiratory rate. So we went from talking about percent oxygen to partial pressures of oxygen, which I'm not going to really get too deep into right now. But I put this in here because if you look here, mitochondria, if they're exposed to 10 to 13 millimeters of mercury for more than 30 minutes, it caused a 50% decrease in mitochondrial output of energy. So if it's lower than that, you would expect even further declines in ATP utilization. Just put that in your brain for a minute. Mitochondria require oxygen for aerobic production, and in vivo, mitochondria are routinely exposed to oxygen tensions in the 2 to 20 millimeters of mercury range. When oxygen tensions fall below critical values, estimated to be about 1 to 2 millimeters of mercury for isolated mitochondria, oxygen utilization by cytochrome oxidase in the mitochondria electron transport chain becomes limiting. Accordingly, oxygen respiration rate should be dependent on the changes in oxygen tension above PO2 critical, but decrease when oxygen tension declines below that. So it's saying here that you're having a 50% reduction in rate in vivo of these cardiac myocytes if it's 10 to 13 millimeters of mercury. So the heart muscle mitochondria, because they're so dense with mitochondria, probably require a higher amount than some other tissues do. But they're saying that the critical oxygen tension is less than 2 millimeters of mercury. Let's go back to this paper here and let's look at some of these percent oxygen and also PO2s of some of these cancers. So we see here the median amount of oxygen average of a percent oxygen is 1.4%, which is well below physiologic hypoxia, and it's into the pathologic hypoxia. Now that's like the tumor as a whole. There are more hypoxic regions within the tumor, and there's more normoxic regions within the tumor. This is like the average of these particular tumors, right? These are not the entire picture. Median percent, right? Median PO2 is 14 to 16 in lung cancer specifically. Now it has all the different cancers here, which I thought was very fascinating, the different oxygen amounts in each of these tumors. And the conclusions of that paper, it said that in experimental studies, it is crucial that investigators control for oxygenation to mimic as far as possible the significance of low oxygen levels found in most human tumor cells, that is at or below 1% and often below 0.1% oxygen. So they're saying the vast majority of human tumor cells is at less than 1% oxygen and often less than 0.1%, down to 0.75 millimeters of mercury, all the way to 7.5 millimeters of mercury. So very hypoxic. Is that fair to say? Most of these tumors are extremely hypoxic, have very little oxygen. Now, what it said here in the paper was, given that TICs, the tumor initiating cells, tend to reside in hypoxic. So not only is the tumor hypoxic, but they tend to reside in the most hypoxic regions of the tumor. Given that TICs tend to reside in the hypoxic or poorly vascularized region of the tumor, they may activate, I don't know why they wouldn't, activate hypoxia-inducible factor 1 alpha and pyruvate kinase isoform M2, PKM2, to sustain glycolysis, or utilize alternative carbon sources such as glutamine and acetate for lipid biosynthesis. So let's unpack that for a second. So we've talked about this in the past. Hypoxia or pseudohypoxia, which I really would like to do some more work on because I think this is a very missed part of cancer maintenance and growth. But anyway, that being said, hypoxia or pseudohypoxia activate hypoxia-inducible factors. Hypoxia-inducible factors are responsible for basically the growth and maintenance of cancer. Now, we've looked at it a lot from the metabolism perspective, and that's where because this is a cancer as a metabolic disease, as a mitochondrial metabolic disease playlist, and that's what the series is called. Of course, that's where we focus on, but it doesn't just only affect metabolism. It affects all kinds of things within the tumor microenvironment. But let's highlight the metabolism. So you see here that HIF-1 alpha, the black lightning bolt, and we've seen this graphic several times before, will upregulate glucose transporters. It will upregulate all enzymes responsible for glycolysis. It will upregulate lactate dehydrogenase A. And then the red lightning bolt is induced by hypoxia-inducible factor 2 alpha. And that is glutamine transporters, glutamine utilization enzymes such as GLS, this is where Don blocks, for example, GLUD, as well as fatty acid synthesis. So keep that in mind that hypoxia-inducible factors, not only do they upregulate glucose and glutamine utilization, but they also upregulate fatty acid synthesis, or the making of new fatty acids. What else does HIF-1 do? HIF-1 also will get rid of extra mitochondrial mass. So for example, if hypoxia-inducible factor is induced or stabilized, it will cause pathologic disease amounts of mitophagy, the recycling of mitochondria, and it will make the cancer cell more dependent on glucose because less oxygen is available. It's assumed that less oxygen is available. Because less oxygen is available, HIF-1 will be stabilized under hypoxia. What else does HIF-1 do? HIF-1, as alluded to by the paper itself that it's in question, will basically shut down electron transport. And then what it's trying to do is, because of the hypoxia, they're trying to protect itself from reactive oxygen species. So HIF-1 alpha will shut down electron transport. So keep that in mind. Now we're back to the paper in question. Results: induction of a metabolic switch by TICs to utilize ketone bodies during glucose starvation. Lung TICs promote cancer initiation and contribute towards resistance, whereas their non-tumor initiating counterparts drive the rapid proliferation of tumor mass. Using a TIC-enriched patient-derived cell line, TS32, referred to as lung TICs, established from the resected primary adenocarcinoma, we also generated an isogenic, differentiated, and less tumorigenic anchorage-dependent line, ADH. Lung TICs were maintained in vitro as tumor spheres, TS, and were consistently enriched for TICs, 90 to 100% with CD166 positive expression. The tumor initiating frequencies were validated and shown to be stable across several passages. Xenografts formed from TICs resembled non-small cell lung cancer-derived tumor tissue and exhibited comparable CD166 plus fractions, approximately 10 to 15%. The expansion of bulk tumor cells is dependent on the access and uptake of glucose to fuel biosynthetic needs. Consistent with this principle, differentiated non-tumor initiating ADH cells were abundant in glycolytic intermediates such as glucose, fructose 6-phosphate, and fructose 1,6-biphosphate when compared to their isogenic TIC counterparts. Because of lower levels of glycolytic intermediates, we reasoned that glycolytic flux might be lower in TICs. Indeed, measurements of extracellular acidification rate confirm this observation. Conversely, the oxygen consumption rate by mitochondrial stress showed that TICs had significantly higher respiratory rate when compared to their non-TIC counterparts. Now, one thing that I was unable to find, and if anybody is better at reading scientific literature that knows, more of the research side of things, I would be forever grateful if you would tell me what you were able to find on this. But basically what they're saying is these TICs had higher mitochondrial consumption rates and higher mitochondrial respiratory rates when compared to their non-TIC counterparts, which again, I cannot make sense of because the TICs tend to be in hypoxic or poorly vascularized regions of the cell. So therefore, the oxygen percent is extremely low. And we know that when oxygen is low, then mitochondrial respiratory rate should decrease significantly. So what I can't understand by looking at this paper is I don't know if these TICs were in a hypoxic environment when they were testing these things. I don't know if they were just in a normal 21% oxygen environment and they weren't controlling for the oxygen, because that doesn't make any sense. If less oxygen is available, how could oxidative phosphorylation going on? I'll digress. The results suggested that TICs might be less dependent on glucose, but oxidize alternative fuel sources, oxidize again, suggesting that these are being fully oxidized through the mitochondrial electron transport chain under hypoxic conditions, but oxidize alternative fuel sources to meet energy demands. To investigate this possibility, TICs and non-TICs were cultured under progressively lower glucose concentrations in deprivation tests. At 5.5 millimoles low glucose, corresponding to fasting conditions in human serum, which we're going to debunk in a second. 5.5 millimoles is not corresponding to fasting conditions in humans. Growth of TICs was unaffected and comparable to standard high glucose cell culture media. By contrast, the growth of ADH cells was severely stunted when glucose was limited, thus supporting glycolytic flux measurements that pointed towards a differential dependency on glucose between functionally different cell types. Again, I'm trying to make the point of that these TICs are in the lowest oxygen portions of the cell. They're upregulating hypoxia factor. They're upregulating the machinery for glycolysis. And then the paragraph right here, they're saying that they are utilizing OXPHOS and not as dependent on glucose. I'm going to go back to the Seafried's paper from April of 2024. The question assumption number 1. Oxygen consumption is an accurate marker for ATP production through OXPHOS in cancer cells. It says here, Winehouse made similar assumptions emphasizing that not all cancer cells had reduced oxygen consumption and could therefore have insufficient OXPHOS compared to normal cells and tissues as Warburg claimed. The same group, Winehouse, later claimed that Warburg's error in thinking that OXPHOS was lower in cancer tissue than normal tissue was due to hypoxia and the thickness of Warburg's tumor tissues slice preparations greater than 400 millimeters. However, earlier findings by Nelsia Mayer showed that oxygen consumption was lower in both tissue slices and homogenates of tumor tissues than the normal tissues and homogenates, thus ruling out slice thickness as a compelling argument against Warburg's conclusions. Moreover, claims of unimpaired OXPHOS function in tumor cells based solely on short-term respirometry, less than 2 hours experiments must be viewed with caution, according to recent findings of Daraj and others. Indeed, changes in oxygen consumption under specific mitochondrial targeting, such as oligomyosin-linked oxygen consumption rate, are subject to many caveats and are not functionally informative on the sufficiency or insufficiency of OXPHOS for viability and or proliferation. It is important to emphasize that the reliance on oxygen consumption rate as a marker for OXPHOS efficiency in tumor cells is just as ambiguous today as it was for Warburg 100 years ago. We recently used novel experimental design in measuring OCR in tandem with ATP-dependent bioluminescence to show that oxygen consumption is not a reliable measure for ATP production through OXPHOS in mouse and human glioma cells. While many recent studies have assumed that OCR is an accurate measure of efficient OXPHOS, none of these studies measured OCR in tandem with ATP-dependent bioluminescence or considered mitochondrial substrate-level phosphorylation as a second OXPHOS-independent source of ATP production in tumor cell mitochondria. Hence, caution is needed in assuming that OCR is an accurate measure of ATP production through OXPHOS in tumor cells utilizing short-term respirometry and without considering glutamine-driven mitochondrial substrate-level phosphorylation as a compounding variable for ATP production. Data from several studies also showed that oxygen in the electron transport chain can be used for metabolic activities other than ATP production through OXPHOS in tumor cells. Oxygen-derived reactive oxygen species, which reduce OXPHOS function and contribute to nuclear genomic mutations, are generally greater in cancer cells than in normal cells. Excess ROS in conjunction with proton leakage can also uncouple the electrochemical gradient, thus reducing ATP production through the ATP synthase. New studies now suggest that ATP production through OXPHOS is neither necessary nor sufficient for brain tumor cell growth in the absence of fermentable fuels. Indeed, no evidence has been presented that any tumor cell can grow in the absence of fermentable fuels regardless of the oxygen consumption. When viewed collectively, the available evidence challenges the assumption that oxygen consumption is an accurate marker of ATP production through OXPHOS in cancer cells. So let's look at what they were talking about in this particular graphic, where they took these TIC cells, the tumor initiating cells, and the non-tumor initiating cells, the bulk cells, and they fed them varying amounts of glucose. So we have here on the top left, figure B, and we see here that there are increasing amounts of glucose given to these TIC cells, and then there are two different amounts given to these ADH cells. So the TIC cells are given 0, 1.4, 2.8, 5.5, and 25. Now remember, they said that 5.5 is equivalent to fasting in humans. Well, 5.5 is equivalent to roughly 100 milligrams per deciliter. So if you are fasting and your glucose is 99 to 100 milligrams per deciliter, you are insulin resistant likely. You likely are not able to get your glucose down to the level that ketogenesis is likely going to happen anyway. So I would highly rebut that. So let's go here. So if you get these TIC cells, 0 glucose, they don't grow at all. Imagine that. If you give them 1.4 millimole, they do grow, although after several days, they do start to even slow down. 1.4 millimole would be equivalent of 25 milligrams per deciliter. That is not a possible blood glucose for humans unless you are giving exogenous insulin in addition to being in a fasted or a highly ketogenic state. 2.8, which is the yellow, also did show growth, which was at 50 milligrams per deciliter. And then 5 point, the only thing we got between there was 5.5, which was the purple line. And then the black line was 25, which is a glucose of 450, which we obviously would see rapid growth with giving someone 450 milligrams per deciliter of glucose. Now, let's look at the ADH cells. ADH cells, they were given 5.5 millimoles, so that's basically 100 milligrams per deciliter of glucose, and they had very poor growth, similar to that of the 1.4 after the 6 days with TIH cells, or the TIC cells. And then they were given 25 millimoles, and that's equivalent to 450 milligrams per deciliter. As you would expect, you would see much higher tumor growth, but still not as high as the TIC cells. So I think it is fair to say that these ADH cells are more reliant on glucose. There's no question. But giving more glucose, does grow these cells more. And unfortunately, in physiologic ranges, 99 down to 50 did have a significant drop. It went from about 1,500 on day 6 to about 1,000. So, 500 cell drop just from going from 99 to 50. So they are very glucose dependent, despite what these authors are trying to push, at least. And then they gave ultra low glucose diets, which was 1.4 millimoles. So they gave a very low amount of glucose to these cells, and they gave lactate as a way to try to salvage these cells. And you can see that the TIC cells could not use lactate as a secondary fuel source. So that was debunked. So the next part of the paper is they talk about the upregulation of ketone machinery by these TIC cells. And basically what they did was they found that when you had high glucose, the TIC cells did not express many of the ketolysis needed enzymes, the BADH1, OXCT1, ACAT2, etc. And then when you put them in an ultra low glucose, which was about 1.4 millimoles of glucose, you would see skyrocketing amounts of ketone body utilization enzymes. Now, we've seen this in papers in the past. As a matter of fact, when we went over can cancer cells use ketones for energy because of the paper that was put out not too long ago about pancreatic cancer cells, we saw a very similar thing that would happen. And this is not really a big deal for us. What this means is that it's going to break down ketones as a salvage, trying to use them for energy through OXPHOS. But the problem is these are in hypoxic regions of the tumor, so therefore there's no oxygen available, and there's structurally deficient number, structure, and function of mitochondria are decreased. So therefore there is structural differences that make that you cannot really effectively burn ketones because ketones cannot be fermented, which we'll talk about. And there's no oxygen to couple these OXPHOS reactions. So therefore they will be broken down. That is clear. And I don't see it as being a big deal for us. I'm going to finish this video here because we've covered a lot already. And as a matter of fact, if I go back up to our outline here, we've covered about half of what we're going to talk about with this paper. This paper was a really, I don't want to say fun to debunk or to at least give a challenge to, but you have to put a lot of nuance into reading papers like this because they will make assumptions about glucose amounts that are normal for humans. They will, I would bet that almost no one is in any degree of ketosis with a glucose of 5.5 millimoles or 100 milligrams per deciliter. There's just enough glucose in the system at that point where your body will not need to tap into ketones. Insulin will be high enough to where it won't happen, or if it is, it's on a very, very minimal amount. And, we have graphics, later can help, better understand that, so if we're at 5.5 right here on this graphic here, here's glucose. Look at the ketones. They're way down here. Right? So in order to get into ketosis, you have to be at a much lower level of glucose than what these people are saying is a fasted state. There's just a lot to debunk here. Again, I don't want to say it's fun, but I'll say that it is intellectually challenging sometimes to go through these papers with a level head and not to get freaked out by, things that they're saying, that they're assuming, et cetera. It's better just to go through the paper and try to give a rational response to these things. Now, what I'll say is, just as a preliminary gestalt about this paper, there are some things about this paper that are actually interesting for us within the ketogenic, metabolic therapy space that I think we can actually take and use for our advantage. In particular, in part 2, we'll see a lot more of that. I think we're going to see a lot in part two also, where the real flaws of the paper are. So far we've not really seen a lot of flaws other than maybe like the assumption that these TIC cells are using OXPHOS, even though they're in hypoxic regions, or maybe the fact that they're saying that, fasted blood glucoses of 100 is normal for humans. But we have not really hit the major flaws of the paper yet. And I really look forward to presenting those flaws because I do think it will help understand better the ground we stand on by employing a ketogenic diet for cancer as a whole. Now, I don't think anybody, including Dr. Seyfried and his group, are saying that the ketogenic diet is a standalone treatment, and we'll echo that in part two. But that being said, it is a diet that makes the most sense as a foundation while dealing with cancer. If you like videos like this, please like, share, and subscribe. And until next time.